All right, so we're going to begin lecture 4. In this lecture, we're going to cover plasma membranes or cell membranes, membrane transport, and finish off our organic molecules. So in the last lecture, we were visiting lipids, and we talked about the fact that there are 4 major subgroupings of lipids. We finished looking at triglycerides and phospholipids, and now we're going to spend a little bit of time on our steroids and prostaglandins. So steroids and prostaglandins are both cell signalers, so we can look at steroids as being these amazing chemical messengers. They have a very different structure, so you can see that for a steroid, we're looking at a molecule that has 3 6-carbon rings, 1 5-carbon ring, and then a carbon side chain. These molecules can be modified to carry out different functions within the body. So for example, if we start with our base model or precursor of cholesterol, this can get modified into testosterone, estradiol, and cortisol, all 3 of which are very powerful in changing cellular activity, but when we look at the structure, are very, very similar. So small differences in structure can make big differences in how these molecules function. Now, all of the steroids have that uniting lipid characteristic of being insoluble in polar solvents. And this is in fact what enables steroids to diffuse through cell membranes and affect cells at a very intimate level. We can look at aldosterone. Aldosterone is a hormone that's involved in regulating our body's salt concentrations, especially potassium. Aldosterone is released from the adrenal gland, circulates through the blood, diffuses directly through the cell membrane of the endothelial cells of blood vessels, and then directly through the cell membrane of cells that it's going to be affecting, specifically in the kidney, where we can see the steroid is going to move through the cell membrane and then affect cellular activity at the level of gene transcription and translation. So this initiates a cascade that eventually results in new proteins within the cell and new activity occurring within those cells. Now, prostaglandins are also going to be cell modifiers. They're going to change cellular activity, but their structure is actually more similar to our phospholipids. In fact, they're going to be produced from phospholipids. The structure is going to be a 20-carbon fatty acid chain that's going to be made cyclical as part of the synthesis of the prostaglandins. So this is a nice structural rendition of what one of those prostaglandins might look like. Like our steroids and all of our lipids, prostaglandins are no different in the fact that they're going to be insoluble in polar solvents or water. These guys are produced essentially by all tissue in the body, which, although the name is prostaglandins, reflects the fact that it was first identified in prostatic fluid. The reality is that these molecules are produced and have effects all over the body. Mostly their effects are going to be locally. So we look at prostaglandins being produced by a cell and then affecting either a cell adjacent or in fact the cell that was producing it. So we have paracrine and autocrine regulation. So autocrine means that the prostaglandin gets released and self-regulates. Paracrine means that the prostaglandin gets released and regulates a cell adjacent. Mediation is usually going to be things like inflammatory response. So prostaglandins can mediate pain, swelling, and fever, as well as just normal tissue function. So blood clotting, gastric secretion, vasodilation, bronchial constriction, uterine function. These are all activities mediated by prostaglandins. If I look at a simplistic example of the pain pathway, here I can see the nervous system initiating the production of a prostaglandin. This is the cascade of resulting in the production of a prostaglandin, which I can see here. We're going to notice that that prostaglandin, once it's been synthesized, gets released from the cell, comes back, and then actually binds a receptor on the cell membrane, and then initiates a cascade, resulting in a regulatory unit that affects gene transcription and production of new proteins. So here again, we're going to find that prostaglandins are going to have local activity, so either paracrine or autocrine, and this will affect the cell's activity. All right, so this is a drawing showing essentially the synthesis and just how diverse the effects can be for prostaglandins. So we're actually going to start with the phospholipid bilayer. We're going to be utilizing an enzyme that cleaves off one of those hydrocarbon tails and then modifies it into a variety of different kinds of prostaglandins. But again, we're going to see that the prostaglandins have mediated effects all over the body. So things like bronchial constriction, bronchial dilation. We can look at the effect on the immune system. We can look at effects on the kidney, and so on and so forth. So briefly, let's look at the biosynthesis of prostaglandins. The precursor model is in fact the phospholipid. We're going to be pulling phospholipids directly out of the cell membrane in order to produce the prostaglandins. There's an enzyme called a phospholipase, which is going to cleave one of the hydrocarbon tails right off of that phosphate head group, hence the name phospholipase. You can see it's an enzyme, so ASE. We're looking at a lipid cleaving enzyme. And the product of this is going to be a molecule called arachidonic acid. So that's the primary precursor for our prostaglandins. So this is a nice drawing here of a classic phospholipid. So remember, you have the polar head group and the hydrocarbon tails. Phospholipase A2 is basically going to chop one of these guys off, and then this hydrocarbon tail will get modified into a variety of different prostaglandins. So here's a more detailed rendition of the phospholipid structure. Again, we're going to target one of these hydrocarbon tails. We're going to produce an arachidonic acid that then gets modified. So if we look at the synthesis of that, again, the primary precursor for our prostaglandins is going to be this arachidonic acid. So here you can see phospholipid. There's our phospholipase enzyme. It cleaves one of those hydrocarbon tails. So here's our arachidonic acid. And then this gets modified. So remember, you have a hydrocarbon tail that has one cyclical pentose ring. There's another enzyme in this process called a cyclooxygenase enzyme that takes that hydrocarbon tail and creates that ring structure. That's called a COX enzyme, or a cyclooxygenase enzyme. And this then modifies it into a variety of different prostaglandin structures. So each of these is going to have a very different specific activity on cells. So you can see here just a short list of some of those, like vasodilation, platelet release reaction, vasoconstriction. We can look at thrombolytic aspects of it, and so on and so forth. All right, so again, we're going to see that the prostaglandins, once they're produced, are going to have a wide range of different effects. So let's just look really quickly at some of the clinical significances for our prostaglandins. Many of you have heard of NSAIDs, which are non-steroidal anti-inflammatory drugs that we can use to help alleviate some of the symptoms of inflammation and pain. The primary mechanism of action is going to be to inhibit that COX enzyme. So once we get arachidonic acid produced, it has to be made cyclical. And if we can inhibit the enzyme that does that, then the hydrocarbon tail does not get modified into the active prostaglandin. And the function of this is primarily to reduce the pain and inflammation of one of the prostaglandins. We also know that prostaglandins have many different kinds of effects, one of which can be clotting. So we're going to find that prostaglandins can be taken, for example, to inhibit clotting. So if you're at risk of blood clots, if you, for example, have atrial fibrillation, you might be on a blood thinner, and one of those may be something like aspirin that affects the clotting reaction. So examples of non-steroidal anti-inflammatories are aspirin, ibuprofen, and Tylenol. So I thought I'd do a quick little video that shows the mechanism of action of aspirin, so we can relate this to prostaglandin action within the body.
Have a fever? Maybe a headache. Many people choose to take acetylsalicylic acid, or aspirin, for relief of their symptoms. In fact, aspirin has been used by humans for more than 2,500 years, ever since some person realized that chewing willow leaves seemed to treat their discomfort. This is because willow leaves contain salicin, which is turned into salicylic acid in the body. Chemists such as Charles Frederick Gerhardt and Felix Hoffmann later experimented with salicylic acid to find ways of making it last longer and become less toxic in the body, which led to the creation of aspirin. Most people have experienced and appreciated firsthand the soothing effects of aspirin, but how does it actually work? In this episode of Medicurio, we will discuss the mechanism of aspirin, its side effects, and other uses of this drug aside from fever and pain relief. The body has many ways to fight an infection. It could raise the temperature and cause a fever, making the environment inhospitable for any pathogen invading the body. In addition, immune cells in the body are stimulated to release a wide variety of toxic chemicals that can kill any invaders, as well as chemicals that stimulate more immune cells as backup, a process known as inflammation. Of course, the higher temperature and released chemicals also damage your own cells, which is why you may feel pain during a fever. Despite this, fevers are not necessarily a bad thing. It is a natural and usually effective way for your body to fight infection, and aspirin and other fever-reducing drugs should only be used if the fever has lasted many days or the body temperature is very high, both of which increase the risk of permanent damage to the body. So what tells the body to turn up the heat, or the immune cells to release their chemicals? Nobody really knew for many years, even after aspirin began to be sold. In 1971, more than 70 years after aspirin first entered the market, British pharmacologist John Vane discovered that a family of fat-derived molecules known as prostaglandins seemed to be responsible for causing fever, inflammation, and pain, and that aspirin treated these symptoms by preventing production of prostaglandins. For his breakthrough discovery, he was awarded the 1982 Nobel Prize in Medicine, which he shared with two Swedish biochemists, Bengt Samuelsson and Sune Bergström, who also did extensive research on these prostaglandins. How does aspirin prevent prostaglandin production? Understanding this requires figuring out the synthesis pathway of prostaglandins, which was accomplished a few years after Vane's discovery. Prostaglandins are derived from fatty acids in the cell membrane. These fatty acids are cut off of membrane lipids by an enzyme known as phospholipase A2. Afterwards, an enzyme called cyclooxygenase, as its name implies, creates a ring in the fatty acid and adds some oxygens to form an intermediate prostaglandin, which is further modified into a variety of different prostaglandins with an incredible diversity of functions. These functions include acting on the hypothalamus to increase body temperature, stimulating immune cells in inflammation, and sensitizing nerves to pain, as well as initiating clotting in blood, constricting and dilating blood vessels, producing mucus in the stomach to protect the stomach lining from stomach acid, and so many more. What aspirin does is that it stops cyclooxygenase from working, which blocks prostaglandin synthesis and results in temporary reduction of prostaglandin-induced pain, fever, and inflammation, until cyclooxygenase activity returns to normal. This inflammation doesn't have to just come from infections. Inflammation from some autoimmune diseases such as rheumatic arthritis can also be relieved using aspirin. However, since prostaglandins aren't just involved in discomfort as mentioned earlier, using aspirin also stops other body functions as well. Sometimes this is a surprising benefit. For example, one of the prostaglandins that aspirin prevents from synthesizing is thromboxane A2, which is involved in activating platelets to initiate blood clotting. This means by taking a low dose of aspirin every day, it becomes harder for blood to clot. This is extremely useful in preventing diseases caused by blood clots, such as heart attacks and stroke, and low-dose aspirin is actually recommended for older-aged people who have a higher risk of suffering from those diseases. On the other hand, aspirin also decreases production of protective mucus in the stomach, which means that stomach acid can burn through the stomach, leading to gastric ulcers and dangerous internal bleeding. Since there is also less clotting occurring, this side effect becomes even more dangerous.
All right, so what we're going to do now is switch gears and move into the plasma membrane. So looking at the cell membrane. At this point, we can start to use all the information that we've learned about organic molecules in order to build our first sort of collaborative structure, which is the phospholipid bilayer. So in this case, we're going to start off with what are the compositions or what are the components of this plasma or cell membrane. And we're going to find out that the primary component of all cell or plasma membranes are going to be phospholipids. Embedded within the phospholipid bilayer, we're going to find proteins, which add some additional function to the cell membrane. Things like cell channels and receptors, anchors and motors. We're going to change the fluidity of the membrane by adding cholesterol, and then provide some additional function by layering in glycoproteins and glycolipids to further enhance things like cellular anchors and cell signals. So this is a nice GIF kind of showing us this phospholipid bilayer. Remember, the phospholipid bilayer, or the cell membrane, like you've learned before, is an amazing structure that defines the identity of every single cell. If I were to say, please draw a cell, I can almost guarantee that students would start off with drawing some kind of circle or square or box, because they are defining the boundary of that cell and defining the beginning of the identity of the cell. The cell membrane is extremely important because it is the beginning point for homeostasis. The ability of all cells to begin to regulate their internal environment different from their external. So this simple line represents this very unique and structured membrane that has in it all these amazing additional features which provide functionality to the membrane itself. So what we're going to do is look at a quick video exploring what we refer to as this mosaic of structures within the phospholipid bilayer, and then start looking at things like membrane transport.
In this video, we will be discussing the structure of the cell membrane. When scientists looked at the selectively permeable cell membrane, they described its structure as a fluid mosaic. You might know that a mosaic is a picture made up of little tiles. Like a mosaic, the cell membrane is made up of different parts as well. The cell membrane has two layers of phospholipids, referred to as a lipid bilayer. The lipid bilayer isn't rigid. The phospholipids in it have the ability to move in a flexible, wave-like motion. Let's take a closer look at a few phospholipids. The round head portions are hydrophilic, which means they are attracted to water. Both the extracellular fluid, meaning fluid outside the cell, and the cytoplasm inside the cell are mostly made up of water. So the hydrophilic phospholipid heads of the outer layer will be oriented toward the extracellular fluid, and the heads of the inner layer will be oriented toward the cytoplasm. The phospholipid tails are hydrophobic, which means watery areas repel them, so they orient toward each other in a direction as far away from the watery content as possible. There are also scattered proteins embedded in the phospholipid layers, some with carbohydrates attached. So, in the fluid mosaic model, the cell membrane is made up of different parts, and these parts make up a flexible boundary around the cell. But how do the majority of substances get in or out of the cell? Some molecules seep through the little spaces in between the phospholipids, which make up the majority of the semi-permeable cell membrane. However, other molecules are too big to fit through the cell membrane this way. So how do these larger molecules pass through the cell membrane? The molecules move through proteins embedded in the cell membrane, either from the extracellular area into the cell or from the intracellular area out of the cell. These substances will move through tunnels made up of these proteins. We'll explore how things move through the cell membrane in greater detail separately.
All right, so this is a still image of that phospholipid bilayer we just looked at. Here we can see the phospholipid bilayer, and then we're going to add into this mosaic of features such as cholesterol, proteins, carbohydrates, which are going to result in an amazing cell membrane that begins the identity of every single cell. If we put it into motion, we can see that the membrane is this fluid structure. So proteins are going to be able to move within it. The phospholipids are going to be able to move within it. So that's why we call it a fluid mosaic model. So we're going to start off looking at the primary component, which of course are phospholipids. Remember, your phospholipids have this amphipathic nature. That means that they will naturally form a phospholipid bilayer if they're put in a water solution. Remember, water is the #1 primary solvent of all living systems. So we're going to find that the phospholipid bilayer will innately, meaning automatically, without having any energy expended by the cell, will automatically form. So to me, that's pretty amazing that it's almost like for a house, you can have the walls self-assemble. And how cool would that be? So cells will literally have their walls self-assembling into these bilayers of phospholipids. Now this is great because that phospholipid is literally going to be the reason that cell membranes have this amazing semi-permeability. If we're going to have a cell that's able to regulate its internal environment, to have an identity different than the outside, it needs to be able to control what comes in and or out of it. And literally the membrane is the beginning of selectively and semi-permeability for all cells, meaning not everything is going to be able to move in or out of them, that the cell is literally going to be able to control what has access to them and begin homeostasis. So with that in mind, remember that the phospholipid bilayer self-assembles because of that amphipathic nature. So we're going to see that the polar head groups form the surface of the membrane, both the external and internal surface. And then the hydrocarbon fatty acid tails form this hydrophobic core. Remember that hydrophobic core is about 40 carbons long because these fatty acid chains are usually about 20 some carbons long. So you have a pretty sort of thick area of hydrophobicity across the membrane. And of course we have our polar head groups that form the surface of the membrane, so the inter and extra surface of the membrane. So again, we have a hydrophilic surface that orients toward water. We have a very long hydrophobic core formed by the hydrocarbon fatty acid tails of all those phospholipids that again are going to self-assemble. They basically twist so that they face away from water. Water, of course, is going to orient around the head groups and away from that water. All right, so again, we notice that because of that self-assembly, we're going to produce a barrier, a cell membrane, that is semi-permeable. So just if all you had were phospholipids and you had nothing else in the membrane all on its own, just because the phospholipids, is what we call semi-permeable. Again, remember, the cell membrane will be the beginning of the identity of the cell. So the cell needs to be able to regulate what comes in or out of it. And the beginning of being able to do that starts with the cell membrane's phospholipids. So the membrane innately, just because of the phospholipid structure, is going to restrict the movement of most substances. There's only going to be a small grouping of substances that can penetrate the membrane essentially without asking, meaning they can move directly through the membrane and the cell has no control over what comes in or out of it. So it's a very, very small subgroup of all of the soluble substances within and around a cell's environment that they can't physically regulate. So let's look at the permeable substances that have free access. So when we say permeable, we mean being able to move directly through the phospholipid bilayer, like we can see here. Because of this cell would have no ability to regulate this molecule moving through, because it is literally just simply displacing the phospholipids and diving through that hydrocarbon sort of barrier of hydrophobicity. So what kinds of molecules are freely permeable? The answer is, like I said, not many. So you have to be something that's small. Why small? The answer is because if you're going to move through the phospholipid bilayer, then you're going to be disrupting the orientation of the phospholipids. Remember, they're sort of standing side by side, shoulder to shoulder, because that is the optimal arrangement in their watery environment. So if you're going to move between them, you have to kind of push them aside. And the molecule is not going to have enough energy or force to displace a large volume of phospholipids, because that displaces a large number of phospholipids, exposing them to water. So they're kind of not physically stuck together, but stuck together in the sense of their optimal arrangement. So if you're small, you're not going to disrupt those phospholipids very much. You'll be able to move through the phospholipid bilayer. So in addition to being small, once you kind of displace the phospholipid bilayer, and this would be if there's a high enough concentration, you're going to be moving into that hydrophobic core. If you're going to move to that hydrophobic core, then you have to also be hydrophobic. So we're going to look at things that are not only small, but also have to be uncharged. Remember, hydrocarbon tails are uncharged, which is why water doesn't really like to be around them. So if you're a molecule that's small and uncharged, you can be able to sit within that hydrophobic core without disrupting the structure of the phospholipids. So good examples of molecules that are freely permeable are things like oxygen, carbon dioxide, and urea. Oxygen, for example, is just 2 atoms, so that's pretty tiny. It's 2 atoms. And they're sharing equally because they're the same atom, so they share electrons very evenly across them, so they are absolutely uncharged. And they'll be able to, if there's a high enough concentration gradient, that'll be the driving force, they'll be able to move across the phospholipid bilayer, disrupting just slightly the phospholipids to allow oxygen access from one side of the membrane to the other. Carbon dioxide is also pretty small. You're looking at 3 atoms. It's just tiny bit polar, so because of that, it's still able, if there's a high enough concentration gradient, to move through that hydrophobic core. And then also urea, which is a nitrogen waste product from protein digestion. This also is a very small molecule that's minimally polar, so it'll be able to dive through that phospholipid bilayer. So these are molecules that are freely permeable, meaning, again, the cell cannot regulate the movement of these substances because they're going to freely go through the membrane. But again, that membrane needs to be able to regulate. That's one of the great things about the membrane. It needs to be able to regulate the movement of most things. So again, we're going to find that only a very small grouping of molecules are going to be small enough and uncharged to be able to have this free access through the membrane. All right, so what other things are going to have free access? There's only one other grouping of molecules that have free access, and we've already kind of looked at those. And those are going to be our lipid-like molecules, so our prostaglandins and our steroids. They'll be able to navigate through that phospholipid bilayer with a minimal obstruction and be able to gain access into the cell. So again, if you're small and uncharged, or if you're lipid-like. So that's pretty much it. This always, to me, is kind of scary, thinking a steroid doesn't have to ask permission to get into a cell. It just literally goes right through. It's one of the reasons for which when you use topical medications, they emulsify them in ointments or creams, because remember, they're going to have that free access through the membrane. So when you think about your skin's cell membranes, they can basically move right through them and then eventually get absorbed. All right, so if those are the things that can get across, what are the things that can't get across? And then how do we allow cells to have access to the impermeable substances? So the impermeable substances, meaning molecules that cannot freely move across a membrane by displacing the phospholipids, is actually a very large number of molecules. So things that do not have free access are things that are large and nonpolar. So even if you're not charged, but you're too big, you can't displace the phospholipids to a large enough degree that they'll be able to have free access. So when we say large doesn't actually, in my mind, have to be very big. So for example, glucose, which we know is only 6 carbons, 12 oxygens, and 12 hydrogens and 6 oxygens. To me, that's not a very big molecule. But that molecule, even though it's uncharged, is not going to be able to displace the phospholipids. It causes too much of a disruption to be able to displace those phospholipids and freely diffuse through. So large nonpolar molecules, things like our carbohydrates and proteins, do not have free access. So those are 2 big organic molecule groupings that do not have free access. Also, any of our charged substances. So those are going to be things like sodium, potassium, chloride, calcium. Those are not going to have free access. So even though they're tiny, I mean, sodium is 1 atom, it is not going to be able to navigate through that 40 carbon long hydrophobic core because it's charged. I mean, if you're thinking about it, the driving force is going to be a concentration gradient. And if sodium is going to move through, it has to pass through all of that hydrophobicity. And it would much rather have stability in a watery environment than be forced through a hydrophobic core. So charged entities, ions and ionic molecules, are not going to be able to diffuse directly through the phospholipid bilayer. And then molecules that are significantly polar. So even though they're not fully charged, if a molecule has significant displacement of charge within the molecule, it also is not going to be able to diffuse through that hydrophobic core. So water, although maybe some of you will be surprised, does not have free access in most cases. It has to have a really, really high concentration gradient if it is going to move through that hydrophobic core. And in most cases, water is going to need to have assistance to get across. So we actually looked at those when we looked at proteins. The assisting molecules are those aquaporins. So they will literally be protein channels. So here's a drawing or an animation showing us how molecules that otherwise don't move through the phospholipid bilayer gain access into the cell. And when cells make proteins, proteins, remember, are the tools and the machinery of the cell. They define what the cells are able to do. In this case, when we're looking specifically at the membrane, and the role of the cell at this point is going to be maintaining homeostasis, defining its internal environment, we can see the cell is able to do that by producing specific proteins that will then sort of shepherd across the membrane exactly what that cell wants. This is a really great animation where we're looking at basically the phospholipid bilayer. In green, these are the hydrophobic hydrocarbon tails of the phospholipids. So these are all the fatty acid chains. The red circles represent phosphate. So this is where the phosphate head groups of the phospholipids are. And the little blue spheres represent water. And I think it's really cool to look at when water is represented. You can see that water clusters all around the phosphate head groups, but doesn't travel into this hydrophobic core. Remember, even though water is a pretty tiny molecule, because of its significant polarity, it doesn't have enough force to move it across this 40 carbon wide hydrophobic core. All right, so to summarize this, the membrane is going to restrict most movement. So we can see here lipid-soluble molecules, so of course this is our cholesterol, can diffuse through the membrane. So if you're lipid-soluble, you can diffuse through. If you're small and uncharged, you can diffuse through. But if you're large, and you can see here, here's a glucose monomer, or at least a monosaccharide. I don't quite know which one it is, but a monosaccharide, you can see it bouncing off because it's too big. And then this, of course, is a protein. So again, if it's larger than, a few carbons long, we're going to find that it's not going to have access. So these are considered large molecules. So when we look at carbohydrates and we look at proteins, those organic molecules are not going to have free access. So all we really have having free access are going to be our lipids. If you're ionic or charged, you also are not going to have free access, and they're going to need assistance to get across the membrane. And this is drawing one of those protein sort of channels or shepherds that allows access for these charged entities. All right, so that's the phospholipid bilayer. So when we're talking about the cell membrane, remember the phospholipid bilayer is going to make the majority of it, and just by having that phospholipid bilayer, we have this amazing barrier, this semi-permeable structure that's going to permit very few things to have free access and restrict almost everything else. So restricting the movement of most stuff. So let's go ahead and look at our proteins, which we know are going to be important as channels, but other functions within this membrane. And then we'll spend some time looking at how the proteins are going to be moving molecules. All right, so integral membrane proteins are proteins that are permanently associated with the membrane. So remember, we're looking at membrane structure. The next component of the membrane after our phospholipids are the integral membrane proteins. So these are proteins that will afford additional function to the membrane itself. So sometimes that'll be to anchor. We can see here we have proteins that traverse the membrane from one cell to the next. This is going to anchor these two cells together. So that is an example of one of these integral membrane proteins having an anchoring role. But it also can be like, here's a vesicle. These may be neurotransmitters, and we may want to move them. We're going to use our integral membrane proteins here to allow for an anchoring and then binding to the membrane. So we're going to talk about these structures a little bit later as part of the nervous system. So integral membrane proteins are going to play a very important role for how the membrane will function, not just in anchoring, but also in binding. Integral membrane proteins will also play a role in channels that we looked at with the movement of molecules that otherwise are impermeable. So potassium, for example, is a charged entity. Normally, even though it's small, cannot diffuse through the phospholipid bilayer, and therefore has to essentially ask permission to travel through by going through a protein that the cell will actually make. And we can also see proteins over here having a role in communication or cell signaling. So this is an acetylcholine. It's a neurotransmitter. It's able to change the activity of the cell by binding with a receptor in the membrane. So it's almost like a doorbell at the surface of the cell, sort of requesting access and asking for permission to change the activity inside the cell. So we'll look at receptors in a lot of detail later as well. So here's an example of a protein that actually is functioning both as a receptor and a channel. Acetylcholine is in fact a molecule that'll be really important for muscle contraction. In fact, all skeletal muscle is going to be stimulated by acetylcholine binding to a channel that looks just like this one, and then causing the movement of charge and initiating action potentials. So we're going to spend a lot of time talking about this process. All right, so here's a quick GIF showing us, or actually I think it's a video, showing us sort of signal transduction. So here's the phospholipid bilayer. We can see a protein embedded in that membrane. We can see molecules outside the cell that are going to change the activity by binding. So you can see there's an alpha helix, and this is a peptide bonding a very large protein, changing its shape because it's interacting with R groups. It's going to cause a change in shape. And then this will initiate a response on the cell's interior. So again, we can see that proteins in the membrane are going to have many functions, one of which is going to be sensitivity to the outside environment. All right, so and then this is a nice GIF again showing us integral membrane proteins that are going to activate maybe a response within the cell. So this again is a receptor that will then initiate a cellular response, so providing sensitivity. Just a reminder, when we look at proteins associated with the membrane, if they're going to be a permanent part of the membrane, then we need to be able to structure them so that they're always going to be associated with it. And so how do you stick a protein in a membrane? It's actually really easy. All you have to make sure is that the protein has some part of it that is hydrophobic. So here you can see this loop over here of an alpha helix has R groups that are hydrophobic, and this will essentially be the glue that embeds that portion of the protein in the membrane. So if I were trying to remove this protein or pull it out of the membrane, it will resist that because of the hydrophobic R groups on those loops of alpha helices. They're going to have stability within the phospholipid bilayer, and so to remove them would require some force. So that means that normally this protein may move side to side. Right? We can glide through the phospholipids. Remember, these phospholipids are not attached to one another. They're just sort of placed next to one another to sort of orient to their best or optimal position. And so that means that this protein can actually glide in and amongst it because it's not going to interfere with the stability of the phospholipids. Here's another example. You can look at this protein. If we wanted a protein to be stuck in the membrane like this and move only back and forth but not rotate, we can solidify its position by making sure that the protein has hydrophilic portions facing the exterior of the membrane and hydrophobic portions in the center. If this is the case, I wouldn't be able to take this protein and move it. So let me erase my drawing there. I wouldn't be able to take this protein and move it because if I tried to rotate it, it would force that hydrophilic portion of the protein through the hydrocarbon hydrophobic core here. And so this protein would not be able to flip, but it would simply be able to move side to side. So this again is representing that fluidity of the membrane. So these proteins can glide. That'll be really important later when we're dealing with cell signaling. But they're solidified in the membrane simply because of that hydrophobic, hydrophilic structuring of the protein. So that's how we can embed them in the membrane. So you can't pull them out, right? Because that, of course, would mean the hydrophobic portion of the protein would be in water. And that's not going to happen. We can't twist them because that would pull these hydrophilic portions into the hydrophobic portion of the membrane. But they can move laterally back and forth because that allows for the membrane to maintain its stability. All right, so now let's look at cholesterol. Cholesterol is referred to as a fluidity regulator or fluidity modulator of the membrane, meaning depending on how warm or cold the membrane is, it can make sure that the membrane maintains its relative fluidity. So remember, it's all about homeostasis. We want to make sure that the membrane has the same level of movement within it so that proteins can move laterally within it. We can get cell signaling happening. So we need to make sure that the membrane is always the same level of fluidity, regardless of how hot or cold the cell is. And so cholesterol does that. Cholesterol essentially buffers, right? And when we talk about a buffer, like we think about buffers for pH, buffers stabilize. So in this case, cholesterol is a fluidity stabilizer. Make sure that the fluidity changes very little. So I have here, if it's a very high environment around the cell membrane, cholesterol functions to decrease fluidity. If it were embedded in the membrane, it's going to make the membrane less able to move, kind of putting like an anchor in the middle of it. At very low temperatures, cholesterol is enough of a change in shape that it creates a sort of an obstruction. And that means that the phospholipids are not going to line up quite as easily or evenly. And that means that it's going to be an area of movement. So very similar to when we looked at the double covalent bonds. If you have a little kink in it, it's going to make the membrane more fluid in the lower temperatures. So this is a kind of a nice drawing here. We can see here's the phospholipid bilayer. Here we can see a cholesterol embedded in the membrane. And we can see that if it's cold, this creates a disruption. And that means that there's a little bit of a gap. And that means that the membrane will be much more fluid. But if it's really hot and these guys are bouncing around because of the energy and the temperature of the environment, then this cholesterol helps to minimize their movement. So it would be decreasing the fluidity at that point. So that's what's great about cholesterol. It's this fluidity regulator. All right, and then last but not least, we have glycoproteins and glycolipids. Basically, these are either proteins or lipids that have sugar attached to them. And these can play many roles, but things like cell adhesion and cell recognition are two roles that these glycoproteins and glycolipids are going to have. So essentially, they're going to afford more functionality to the membrane. We're going to spend a lot more time on these guys. These are actually red blood cells, and we're looking at the ABO blood typing. And I don't know but to you, but it was super shocking to me to know that the difference between AB, and O is just really the glycosylation of one of these membrane proteins. So these are just different monosaccharide residues, and depending on the glycosylation of a membrane protein here called antigen H, we can actually produce the different blood types. Okay. This case, we're looking at a glycolipid where we have cell surface molecules that are going to be affording the ability of cell recognition and adhesion here. So this is the implantation of a blastocyst into the uterine wall. So this process is mediated by the glycolipids. All right, so with our basic structure of the membrane, we're now going to look at some of the overall functioning, right? So remember, the membrane is really important in allowing cells to have a self-identity. That's because when we think about what the membrane does, it allows cells to define their internal environment different than their external. It provides them ability to physically isolate their internal environment. And remember, what it is the first sort of line of defense against the dysregulation of homeostasis is that phospholipid bilayer. So the phospholipid bilayer we can see here, this is semi-permeable. And so most things are not going to be able to freely pass it. So the cell is going to have the ability to define what moves across based on what it puts in its membrane. So the membrane's function is going to be to physically isolate the interior of the cell from the outside environment and begin this process of defining its own self or its own identity. Okay. All right. So phospholipid bilayers prevent the movement of most things across the membrane. In addition to that, we know that the membrane is going to be part of this regulation of homeostasis. Like I said, the membrane itself, because of the phospholipid structure, is semi-permeable. But if the cell wants something to move across it, it can actually achieve this regulation of what moves across by producing sort of a variety of different protein channels. So depending on maybe if it wants to be positively charged inside the cell, it might make channels specifically for sodium to enter. If it wanted to be negative inside, it could make channels specifically for chloride to enter. So depending on what the cell wants, it will afford unique sort of internal environment by producing particular proteins and embedding them in the membrane. So the membrane will have this ability now to regulate and maintain homeostasis by what the cell builds and inserts in the membrane. In addition to that, the membrane, like the door, or I should say the walls to your home, if you want to be able to know what's going on outside, you have to be able to be sensitive to the exterior environment. So that's why you might need to have a window or a doorbell so you can see what's going on outside. The membrane is the wall of the cell, and so it needs to be able to interact with the exterior environment. The exterior environment needs to be able to talk to the cell, and that's going to happen all through that cell membrane. The cell membrane is what's going to provide the sensitivity of the cell to the outside world, so it can be able to respond to respond to the signals in the environment. Will have receptors. So here you can see acetylcholine released from the nervous system is going to be able to communicate with this cell through a receptor that the cell makes in the membrane. Obviously, if this receptor is not here, this neurotransmitter would then be silent. So the cell gets to decide what it listens to from the outside environment by what it puts in its membrane. All right, so here's a nice GIF showing that. Here's a neurotransmitter that's binding. It's going to set up a series of responses. So it's kind of like you can push the doorbell and maybe it turns on a camera that then initiates a response inside the cell. Okay, so just an example of how important this membrane is. And notice that these molecules have to move back and forth within the membrane. So even though I don't know exactly the entire structure of this, I know that this portion of the protein is going to be hydrophobic, and that's why it's able to move back and forth. It's going to bind, so we can see the protein binding another protein and initiating a response. Here's another example This is the cell membrane. These are dopamine neurotransmitters that get released. They're going to diffuse across the synaptic cleft and bind with the receptors in the membrane of the postsynaptic cell. This will then initiate a response in that postsynaptic cell. So when we look at communication between things like neurotransmitters and other cell signaling molecules, they're going to have to interact with the membrane first before they can have a response inside the cell. All right, so now let's look at movement across the cell membrane. So first of all, if we're going to talk about movement across the membrane, we have to talk about just regular movement of molecules. And I know for me, I didn't get introduced to this term Brownian motion until very late in my study of cell structure and function. And so I thought I'd begin by putting in this term Brownian motion, which I think is essential in understanding diffusion, osmosis, and membrane transport. So Brownian motion is this idea that if there's energy in a system, so obviously you have to have energy in a system, that particles will always move randomly, and their movement is always going to be due to basically the bombardment of other particles. So if I'm looking at this animation, I can see all these little spheres, and they're going to be moving in the environment. So they would move in straight lines, but you'll notice that they're changing their position, and the reason they change their position and move in different directions is because of bombardment. They're hitting each other. So Brownian motion is this idea that we have this innate random motion of all matter, and their direction of motion is going to be due to bombardment or the fact they're hitting each other within their environment. The degree of motion, like how much motion occurs for molecules or particles, depends on how much energy is in the system. So the warmer this is, the faster these molecules are going to move, the higher the likely they're going to hit each other and change their direction. So this will be the beginning of our discussion of diffusion.
The air is still, and the bright lights are streaming through the window. What do you notice about the dust particles? The black circles as they pass through the beam of light. Pause the video and think about this. Press play once you've made up your mind. The answer is that the dust particles are moving randomly. Did you see it? The dust particles are not always moving downwards, but are moving in all directions. The dust particles are very small and are supported by a fluid. In this case, that fluid is a gas. You are unable to see the gas particles directly because they are too small, but you can see the larger dust particles. The reason that the dust particles move randomly are because of the gas. We know that the gas is there because every time we move our hand through the air, we feel them against our hand. Try it now. Continue when ready. The particles in the air move in random directions quickly. This means they will have many collisions or will bump into the dust particles many times. Each collision between air particles, very small coloured circles, show how the dust particle, the black circle, moves off in a new random direction. It's a bit like a game of snooker, where there are millions of marbles which collide with larger, observable particles like a ping-pong ball. This effect was first observed by scientist Robert Brown. He was looking at pollen grains suspended in water under the microscope, and he wondered why they jiggled around. He thought that the pollen was alive. It was later that scientists realized the importance of his discovery. Now, here's a challenge for you. Thinking about what we know about dust particles, can you explain why the pollen grains appear to be jiggling randomly? Pause the video and continue when ready. In summary, the fluid is no longer a gas, but is a liquid, water. The pollen grain is colliding with many water molecules. Each water molecule is too small to see, but we can see the combined effect of their collisions on the pollen grain as it moves randomly. This effect is known as, you guessed it, Brownian motion.
All right. So the idea of Brownian motion is that all matter is going to essentially move randomly as long as there's enough energy in the system. All right, so let's look at the first and second law of thermodynamics. We're going to focus on the second law that basically says that the universe is naturally becoming more disorganized. It'd be nice if we kind of organized, disorganized. And if you were to look at just particles and then watch them over time, you're going to see that they're going to bombard each other, collide with each other, and over time become less organized. The degree of randomness or the degree of disorganization is referred to as entropy. So entropy is this idea that we have a system that is becoming more and more disorganized, and the longer we let it sit, the higher and higher the entropy. So all naturally occurring motions function to increase entropy or increase disorganization. So these little bombardments here are going to be pushing things around, and so that they're less and less organized. Now with that in mind, we're going to start to look at how does this work in the body within environments of cells. And we're going to begin with looking at diffusion. So diffusion is a term that you guys have probably spent a lot of time talking about in general bio and in physics and in chemistry. Diffusion is the movement of particles, right, from where there are more of them, right, to where there are less of them, right? There are many ways to define diffusion. That's one. You can say diffusion is the movement of particles, we say, down their concentration gradient. So again, from where there are more of them to where there are less of them. You can also say, in the context of entropy, from where they are more organized to where they're less organized, right? So this is just a natural phenomena of all matter. It's not defining life, because according to the second law of thermodynamics, all systems, if there's enough energy, move to disorganization, right? So again, diffusion just really defines that in the context of particles, and says that if the second law of thermodynamics is true and all systems work to become less organized, then they'll work from a organized state where there's more of them on one side to where there are less of them. They're going to move from an organized to a less organized state, or down a concentration gradient, right? So if we're looking at these particles over time, they're going to move so that they're evenly distributed. When I have them starting off on the one side, wait till this GIF starts again, this is organized. If given enough time, they move across, they become disorganized. All right, so diffusion results in, if we allow a system to run for long enough, equilibrium. So if I'm looking at this system, I start off with more particles on one side of this beaker than the other. This is organized, right? It's taken some energy to make sure that there are more molecules on this side or particles on this side than on the other. If I allow this system to randomly move, I'm going to find that over time, according to that second law of thermodynamics, we're going to move until there's no organization, right? And everything is evenly distributed. So all of the constituents will eventually be evenly distributed if they're allowed to randomly move like we define with Brownian motion, okay? So we would result in this sort of random distribution across the whole space. This is the highest level of entropy, and when we hit this, we're going to find that there's no more net movement of molecules. There's going to be an even movement of molecules back and forth, but we're not going to see an overall movement from one space to the next. So we're going to say that at this point, we have no more diffusion taking place, even though individual molecules are going to be moving back and forth. Okay, so diffusion will always result in the movement of particles from an organized to a less organized state, where we're going to be increasing our entropy. All right, so here's an example of that, right? We're going to look at this GIF. Right now, each of these particles is moving randomly. So each one moves randomly. They move in straight lines until they collide with something. Remember, bombardment changes their direction. And if given enough time, they're going to move from where they're more concentrated to where they're less concentrated. Now, these particles don't know where they're moving. They're moving randomly. Some will move into a higher concentration. Some will move into a lower concentration. If they move into a lower concentration, there's nothing that's going to hit them, so they can track all the way across. And this allows for this spreading out, or this high level of entropy when we're done. All right, so here's a quick video on diffusion.
Diffusion is the net movement of a substance from a region of high concentration to a region of low concentration. Let's use the sugar cube and beaker of water as an example. When the sugar cube is dropped into water, it begins to dissolve. The sugar molecules in the cube move from an area of high concentration to areas of lower concentration until the molecules are evenly distributed. This type of motion is known as diffusion.
All right, so again, diffusion is really just describing Brownian motion in the context of organized to disorganized state. So let's look at a clinical significance of why diffusion is so very important, and we're going to look in the context of dialysis.
Hemodialysis is a medical procedure. It's one of the ways you can treat severe kidney disease. The way hemodialysis works is that it uses an artificial kidney to replace some of the functions of a healthy kidney.
Blood is taken out of the body, goes through the dialysis machine, goes through the filter, is cleaned, and then it comes back into the person. So you don't really lose any blood. It all goes through the dialysis machine, gets cleaned, and then comes back into the body.
So it's really that artificial kidney and the blood working together to clean the fluid in my body. You're taking that urea out of the blood, you're taking potassium out, the different things you want to balance out of the blood. So then it's like a magnet, and it'll draw those solutes out. It's an amazing process when you think it has to get to the cells all through your body, from your toes to your head. Each one of those cells has fluid in it that needs to be cleaned. Everything you normally would get rid of through urination, you're trying to get rid of through the hemodialysis.
And when people dialyze, sometimes if they do conventional dialysis where they're dialyzing 3 times a week, they may feel sort of tired after they have dialysis, but then after a while they feel better. People who do more intensive dialysis or daily dialysis at home, they feel like they can do things right away.
It isn't that you're living to dialyze. You're going to dialyze so you can go out and live the life you were meant to live.
Yeah, so that's a really great animation. I think one of the things that I think is really important is this drawing right here, where you can see that here's the blood, and these are the constituents we want to remove. So these might be urea particles. The reason they move into dialysate is because of a concentration gradient. So this is really just diffusion occurring between a high concentration and a low concentration. And if you provide that gradient, we're going to see that we can actually move specific molecules from the blood into a waste product that we can then get rid of. So the very essence of what is dialysis really is based on diffusion. All right, so now let's look at membrane transport. So we know that all molecules are going to move from a high concentration to a low concentration. That movement is going to be driven by how much energy is in the system or how hot it is. We're also going to talk later about concentration gradients, but all particles will move from a high to low concentration. So now let's put that in the context of the membrane, and we know that some molecules are going to be able to move directly through the membrane, like oxygen, whereas others are going to have to ask permission. So we're going to expand a little bit more about how we define that movement. We have 2 different ways in which we can move across a membrane. The first is called passive movement, and the other is called active. So passive transport is where the cell invests no energy at all in the actual movement of particles across the membrane, so passive. Active transport is where the cell is going to invest energy in the movement of a substance. So if this substance is simply moving according to a concentration gradient, we're going to refer to that as passive transport. If the cell is going to have to utilize some kind of energy to fuel the transport, then we're going to refer to it as active transport. Okay? So here's a drawing of that. If we're simply diffusing through the membrane, this would be, for example, oxygen, carbon dioxide. This is a form of passive transport because the cell is not utilizing any energy to allow for this to occur. If we have a protein present, right, the cell afforded energy in the production of the protein, but once the protein is there, if we have a channel that allows for molecules to move down their concentration gradients, this is also a form of passive transport. So we're going to talk about facilitated diffusion. And then last but not least, if the cell affords some energy, so in this case ATP, adenosine triphosphate, is actually produced from the breakdown of organic molecules. So the cell is expending energy here. We're going to see the movement of substances is defined by the energy investment by the cell. So this would be an example of active transport. Okay, so let's go back to passive. So passive transport, cells expend no energy at all in the transport of substances. And so there's two ways for substances to get across the cell membrane via passive transport. The first is passive diffusion. So again, this is like oxygen, carbon dioxide, where we're moving through the membrane. The cell is providing no energy in the sense of moving the molecule. So this is purely coming from the concentration gradient that exists. And we're moving through the phospholipid bilayer. So we refer to this as passive diffusion. If the cell has to construct a protein first, right, but then the movement is according to a concentration gradient, meaning the energy is coming from the gradient of the molecules, then we refer to this as facilitated. To facilitate means to help. So the cell has to build the protein first. So it is helping by building a protein channel, but then after that, all movement is going to be passive. Okay? So an example of this would be our aquaporins. So water doesn't normally move through the cell membrane on its own. It's going to need a channel. All channels in the body actually have aquaporins. So water is then going to be able to freely diffuse according to concentration gradients as long as the channels are present. So that'd be an example of what we call facilitated diffusion. All right, so here's a movement of molecules. So this is an example of passive diffusion. I know that because there's no protein in the membrane. And I can see the molecules are moving according to a concentration gradient. All right, so passive transport, again, remember the energy that's required for movement. All things require energy to move, but where that energy is coming from is not the cell. It's coming from the concentration gradient. So remember, according to the second law of thermodynamics, all things are going to move to become eventually more random, and that means they're going to move from a higher concentration to a lower concentration. So all particles will always move from a high to low concentration, and if they're small enough and they're uncharged, they can literally move through the membrane. If there's a concentration gradient but a protein channel, they can also move through the membrane through the channel based on that concentration gradient. So in passive transport, the energy from movement of particles is coming from the concentration gradient. Okay, so here's a nice example of facilitated diffusion. How do I know it's facilitated? I can see the protein. And if it's diffusion, I'm going to know that's diffusion because I'm going to see a concentration gradient and movement from a high to a low concentration. All right, so here's an example of a facilitated diffusion. Notice the high concentration to low concentration through a channel. So if I saw this animation and I said, what kind of diffusion is depicted in this animation, you would have to say facilitated diffusion. The cell is not expending any energy in the movement of the particles. It's relying on the energy in the concentration gradient, but we need a channel in order to facilitate that process. All right, so in this case,
some proteins in a cell's membrane act as channels for specific ions or molecules. These channel proteins don't use energy at all. They simply allow the materials to naturally diffuse from the side with more solutes to the side with less. Whether the direction is out or in depends on where the concentration is higher for each different solute.
All right, so again, that would be passive transport because even though we're using a protein, the cell is not expending any energy. The energy in transport is coming from a gradient. So you can move into a cell or out of a cell as long as there's a channel and a gradient. All right, so active transport is where the cell is actually going to expend energy. So there's energy in the transport. So why would a cell expend energy? What is it trying to do? And if all molecules naturally move down a concentration gradient, that happens naturally, the cell's not going to spend any energy to do that. Then if you're going to go in the opposite direction, where you're going to be building a concentration gradient, you're going to be working against sort of the natural movement of all particles, that's going to require energy. So anytime a concentration gradient is actually made, right, created or increased, that's going to require energy. So here I have a particle in an area of high concentration, right? So if I move across from a high to a low concentration, this is natural, right? Cell doesn't need any energy to do that. But if I want to take a particle and push it up a concentration gradient, that's going to require energy. So active transport, what you're going to look for is a gradient being produced or increased, and the cell is going to expend energy. In this case, it's actually utilizing energy in ATP to allow for this increase in concentration gradient. So in this one, it says energy from ATP is used to move a particle through a protein against diffusion. So diffusion is natural. If you're going to work against it, you're going to have to afford energy to do that. So active transport is always going to be the movement up a concentration gradient. Okay. So in this case, let me move my drawings. Right. In this case, I'm going to see passive transport is down a concentration gradient, and then active transport is going to be up a concentration gradient. Okay. So again, active transport, we're going to be utilizing energy. And there's two primary sources of energy. It's either going to be energy in the form of ATP, like we just saw, or energy in the gradient of another molecule. So we can actually use a concentration gradient of another molecule to allow the movement of a molecule that we're intending. So in all active transport, we're going to see that particles will be moving up a concentration gradient. So from an area of low concentration to an area of high concentration. Okay, so when the transport requires ATP, in this case, we refer to this as active transport and primary. So primary active transport means the energy is supplied by ATP. So that's a primary active channel. In this case, we're moving hydrogen ions. Even though hydrogen's only one atom, technically even just one proton, it's significantly charged, and so it will not have the ability to diffuse directly through the membrane. It needs a protein, and so this is a protein channel, but not just a channel, it's a channel that's going to force hydrogen up its concentration gradient. So that's referred to as a pump. Okay, even though it's a channel, it's an active channel that's utilizing energy in ATP to push hydrogen up its concentration gradient. So this would be an example of a primary active transport. Okay, so here's another example. I have my protein there, and in this case we're utilizing ATP to move sodium up its concentration gradient. So we're going to allow sodium come in, and we're going to move it from the interior of the cell utilizing energy in ATP. It's going to bind, change the shape of that protein, and force it into a different structure where it's going to be open on the outside. This is a great animation because we can really talk about protein structure. Remember that proteins R groups define the shape of the protein, and if I change the R group arrangement, I can change the protein shape. And that's really what's going on here. Sodium is going to bind. That ATP is going to bind also to the protein. It's going to change the shape and force the protein to alter its position from opening up inside the cell to opening up outside the cell. So this is an example again of a primary active transport channel. I know it's primary because it's using ATP, and active because a gradient being produced. So last but not least, we call secondary active transport. This is also referred to as co-transport. It does force the movement of a particle up its concentration gradient. So we're going to see in this case, we're going to be moving sugar from a low concentration across the membrane to a high concentration. So this is active transport. I don't see any ATP, so it's not primary. So you're wondering where is the energy coming from to allow for this movement up the gradient? And the answer is we're going to use the concentration gradient of a different particle to provide the energy of movement. So I have here energy supplied by another substance concentration gradient. And we're going to see this quite a bit, especially in cells where they're going to be moving multiple things. So in the kidney and the intestines, these are two locations where we see a lot of secondary active transport. If we look at the absorption of glucose, it's actually co-transported with sodium. So sodium will provide the concentration gradient to allow for the intake and absorption of glucose. So if we want glucose to come into a cell, we have to have a concentration gradient for sodium. So sodium ends up being higher on the outside. It moves down its concentration gradient, which is simply diffusion. But in that process, it causes enough of a shape change to allow for glucose to move from a low concentration to a high. So we're going to be absorbing glucose. Okay, so one of the reasons why in energy drinks and things like that, it's really important to have not just the sugar but also the electrolytes, because sugar does not have access to the cell unless there's actually some electrolyte in there as well. So that's kind of cool to know. All right, so that's an example of secondary active transport. Here's another little GIF showing us secondary active transport. Again, you're looking at glucose movement. Sodium moves down its concentration gradient, and then glucose is going to be moving up. So we're pulling glucose into the cell. All right, so passive transport results in the movement of molecules down their gradient. So passively, energy's coming from the gradient itself. So if you just looked at a drawing and you didn't have it labeled, you would know that this is passive transport because the concentration gradient is lessening. If we're looking at active transport, again, I can tell, even though I wouldn't necessarily have the label, if a particle is moving up its concentration gradient, I know it's going to be active. And if it's utilizing ATP, then I know it's primary active. So anytime we increase a gradient, it's active transport. All right, and then in this case, this is again showing us the movement of sodium and glucose. This is moving across an intestinal epithelial cell. So you can see next time you eat a bagel or something like that and you're absorbing your sugar, it is contingent upon the presence of electrolytes. So you can see sodium has a concentration gradient. It's higher on the luminal side. It comes into the cell. And then glucose will be moved up its concentration gradient. So this is an example of secondary active transport. Okay, we do need to utilize energy in a primary channel. So if I look down here, I see ATP being utilized. ATP is utilized to actually pump sodium out of the cell. So the reason this works, it's actually kind of cool, is the cell uses energy down here to pump sodium out, which means there's always going to be less sodium in the cell, which means there's always going to be a gradient for sodium. That then allows us to move glucose. So by having a primary active channel down here for sodium, I can actually move both sodium and glucose into the cell, into the body. So I don't actually have to actively transport sodium and glucose. I can just actively transport sodium and then glucose will follow it.
Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient.
All right, so we're going to stop there. I'm going to finish these in our next lecture so we can spend a little bit of time thinking about it without this one being too long.